Now this is a story all about how my life got turned upside down. I'd like to take a minute, just sit right there. I'll tell you how I'm an assistant professor with my own desk and chair. In Southern California, born and raised, on the playground was where I spent most of my days, chilling out, maxing, relaxing all cool, and all shooting some soccer balls outside of the school. When a couple of bosses who were up to no good started downsizing airports in the neighborhood, I got on one long flight and I got scared because Mom said we're moving to a place with blue grass in the air. In Kentucky, I was introduced to entomology, the role of bugs have in society and health. The combination of West Nile virus and male reproductive loss syndrome was all over my University of Kentucky walls. I was amazed that a horse could be killed by something as common as a mosquito or accidentally eating a caterpillar. The perception of entomology is often amusing and frustrating. Entomologists study insects. We raise colonies and catch our prey, but the field is vastly understudied, and what I do is quite smashing. I look, I collect, I analyze, I amplify, I sense, and I omic the crap out of my collections. Entomology is the study of arthropods, those inside-out creatures. They are flexible with their jointed appendages and segmented bodies and scare people with their 6 legs. But not all are scary. Those that should scare us are vectors, because they often bite and transmit pathogens that harm us. Vectors transmit agents, viruses, bacteria, and protozoa, and these are some of the deadliest creatures known to man. These agents are responsible for millions of deaths worldwide, but vectors can also cause blood loss, myiasis, mental psychoses, quarantines, incorrect treatments, and misdiagnoses, and a majority of these actually occur in the southern hemisphere. Mosquitoes and ticks are the best vectors. They have unique biologies and life histories that allow both to succeed. Could you imagine a flying tick, or an immature mosquito feeding on you while you swim? What about finding a mosquito attached to your arm 2 days later? These are just 2 traits that keep vectors in the top 10 of the deadliest animals. Thank you. I whistled for a project, and when it came near, the citizens were being diagnosed with vector-borne diseases far and near. If anything, I could say that this rate was rare, but I thought, can't forget it. A 6-year-old boy died in eastern Tennessee of lacrosse encephalitis within the first year of me being here at the University of Tennessee. Lacrosse virus is transmitted via bites from infected mosquitoes. Symptoms include meningitis and neurological problems in children under 16. The tick-borne pathogens include Anaplasma, Ehrlichia, and Rickettsia, and all cause similar symptoms of fevers and rash. My research program focuses on why these pathogens are in Tennessee and address the questions, from molecules to landscapes. One of the very important, yet very basic themes of my research program is managing vectors and minimizing their impact. Insect management and control is often what the public thinks we spend 100% of our time on, when in fact my program is actually developing new methods and identifying novel modes of action for controlling vectors. This research is often combined with toxicology and physiology to reduce vector populations. Along this line of research, I have teamed up with academics such as Kim Gwynn to evaluate compounds, both chemical and natural, at killing these vectors. Here with an undergrad from Lincoln Memorial University, we identified natural compounds in a native tree's foliage that both controls and repels Lone Star ticks. As everyone in this room probably knows, not all insects transmit the same pathogen. I believe that the foundation of vector ecology program is monitoring and surveillance. And so for the past 2 years, my research program has been focusing on identifying the best methods for monitoring vectors and their respective pathogens. We have been working on improving monitoring methods for both ticks and mosquitoes. We have found that both methods work best for Lone Star ticks and American dog ticks, but dragging a flannel sheet through a field is the cheapest and the best method. For mosquitoes, though, we identified that each mosquito prefers her own trap. This finding will make management a challenge because it will add increased cost and time towards surveillance programs. My tick crew has been working hard in Western Tennessee for the past 3 years. For some reason, hospitalized cases of Rocky Mountain spotted fever leads to significantly more deaths in Western Tennessee than anywhere else in the country. And the fatalities are not likely due to ticks. We were able to identify both Ehrlichia and Rickettsia parkeri in our collections, but have yet to identify Rickettsia rickettsii. But for lacrosse virus, things are a bit different. After the 2012 fatal case in Union County, we not only identified the same composition of mosquitoes in cemeteries as in the case house, but we also identified overlapping population clusters and identical sequences in both the vector and its victim. There is a critical need to identify the risk areas and host vectors. This is the first step for a management program, and it's likely that habitats are influencing the presence of hosts, and in turn, the presence of vectors, and inevitably, the pathogen population. Consequently, I have assembled a team to help identify preferred vector habitats, and as expected, not all vectors are the same. Some are everywhere, while others are quite specific. Lone Star ticks are found everywhere. Gulf Coast ticks are only found in grasslands, and not on the Gulf Coast anymore. And both the American dog and black leg ticks seem to be correlated with distance to water. Also, with the help of Pat Kaiser, it seems like a natural environment gives us the fewest ticks, while burned or managed forest sites produce a larger tick population. Those habitats then influence the vector's host. We have identified ectoparasites of white-tailed deer, cattle, and on small mammals. But most interestingly, the greatest abundance of ticks have been on hosts collected in pine habitats. My program also attempts to identify the genetic traits associated with the dispersal of the vector and its hitchhiking pathogen. Hypotheses to be addressed include identifying the genetic differences between the pathogen and/or the ticks in western Tennessee, which accounts for the increased fatality rate. Some things that I question and I wonder about are, is there a permissive protein or an extra virulent vector that makes this happen? I'm trying to identify the ideal genes and genotypes involved in vector transmission. To do this, I've partnered with Jennifer DeBrun to identify microbial communities within our vectors, to identify potential synergistic or antagonistic bacteria associated with our pathogens. Thus far, we've discovered differences among engorged and questing ticks, as the blood meal appears to alter the community dynamics, and microbial communities differ. I hope I brought some light into the importance of understanding bugs to improve and save lives. I pulled up to the campus in March of 2012, and I yelled to my mentors, "Yo, Holmes, I'll call you later." I looked at my university. I'm finally here, a real assistant professor with my own desk and chair.